Cloning sounds like something that's probably very complicated and expensive, which in reality it may be, but I'm going to break it down so you understand how cloning works in a basic procedure. First, we take the somatic or reproductive cells from an adult that was to be cloned. Somatic cells come from the body and are not gametes like sperm or egg. Then we remove the nucleus from the cell, and this is called enucleation. The nucleus that we removed is then placed into another enucleated donor egg. So again, enucleation removes the nucleus in order to maintain the appropriate chromosome number. This new cell is then stimulated with an electric shock, and embryo development begins almost immediately, as if it were a normal zygote. This results in an embryo that is implanted into a surrogate mother. So again, remember, we implant the embryo into a mother who didn't technically have those genetics, but is growing that cell or growing that clone simply because the other animal has reproductive challenges or complications. So the diagram over to the right kind of explains how this works too. We remove the nucleus from the egg cell. We take the somatic cell or the somatic body cell with desired genes. We combine them with an electric shock, and then this creates a clone in our surrogate mother. This is how essentially Dolly the cloned sheep was cloned in 1997. It says in 1996 here, but she wasn't actually born until 1997. I'm not going to read this. It's basically just what I said, but just written out a lot more extensively. But if you're interested, feel free to pause and learn a little bit more about Dolly the cloned sheep. And we will stop there. In our next video, we're going to talk a little bit about artificial insemination, or AI.